This talk is actually, in a very real sense, perhaps should have come before the previous one, and it maybe fits in a little better with Dr. Marshall's first talk. The question we ask ourselves is, what happens and why? So again, we'll start with this same cartoon paradigm: injury, followed by an acute stress response, SIRS and CARS, septic shock or severe sepsis, malignant inflammation, immunosuppression, MODS, and resolution and death. Again, the standard model that we believe we've been looking at. Now, once again, it's important to note that maybe malignant inflammation and immunosuppression come before septic shock, maybe not, and we've added in the little element of chronic critical illness. So, once again, 4 distinct processes. We'll run through this very quickly because we talked about it already. The first, where SIRS and CARS lead to inflammation and immunosuppression. The second, where either the inflammation itself or the combination of inflammation and immunosuppression leads to septic shock or severe sepsis. The third, where MODS or leads to chronic critical illness, which we spoke about in the first one. So we're not going to talk about that in this one. And then finally, this particular transition. Why does septic shock or severe sepsis often end up in MODS? What is going on in this transition? What should we think about this, and how will that affect what we think we need to do with our patients? So, once again, the proposition is that septic shock, severe sepsis, and MODS are pathological responses to uncontrolled inflammation. Why do we think that? Well, again, we talked about 2 key problems. This time we're going to talk about the fact that we believe this response to be pathologic. I hope that what I'm going to do is convince you to at least consider that that might not be the case. So, a short history lesson. The first reports of MODS, of organ dysfunction, primarily in the surgical literature after rupture of AAAs or in the face of severe trauma, reported quite some time ago, 73, 77. Typify the emerging clinical picture of patients kept alive solely by reason of specific mechanical and pharmacological support, Ben Eisman's statement in that paper. And by the mid-1980s, the time when I was first getting my start in the critical care world, there was an extensive literature on MODS. They didn't call it MODS, they called it MOSF or MOF or something else, but it was typified by this sort of paper by Goris et al., multiple organ failure, generalized autodestructive inflammation. Again, pathological, based on inflammation. The patients developed multiple organ failure, the outcome was different, it was dismal, and therefore MODS or MOF or whatever we're calling it was a pathological response that was accepted. So the first question we'll ask ourselves in exploring this particular topic is, how does organ dysfunction develop? Again, we go to our little picture, and when we think about it, what are we talking about when we talk about organ dysfunction? We're talking about hypotension, hypoxemia and hypercarbia, decreased urine output, increased creatinine, an increase in bilirubin, the clinical hallmark of hepatic dysfunction, encephalopathy and neuropathy, and we should add myopathy. On a pathological level, on a level of actual cellular function, we're talking about cardiomyocytes that don't contract, or endothelial cells or smooth muscle cells that don't contract properly. We're talking about impaired gas exchange and failure to transit across the alveolar membrane, impaired renal resorption, limited hepatic protein synthesis, and impaired neurotransmission. In fact, what we're really talking about is every cell in the body in some way, shape, or form becomes dysfunctional. What kind of things can cause every cell to become dysfunctional? And the first thing that certainly would come to mind, I would think, would be that there's no energy to do this. The power plant is missing. It's not working properly. Energy, of course, is generated by mitochondria, by and large, and mitochondria, of course, require oxygen. And tissue dysoxia has been very well described in settings of sepsis, severe septic shock, MODS. Where does it come from? Well, there are 2 basic theories to explain the dysoxia that we see in patients with multiple organ dysfunction. The first is that there is microvascular dysfunction, thrombosis and shunting with inadequate oxygen delivery, The second is that there is mitochondrial dysfunction, so-called cytopathic hypoxia, where there's inadequate oxygen utilization. Now, both of these are perfectly feasible, and both of these can be studied, and both of these have been studied. But again, presumed to be pathologic. The study of the microvasculature has taken many forms. This is one that I particularly like. It's Jan Ince's work from the Netherlands. What he's done is looking at submucosal blood flow, and he's defined a whole series of abnormalities involving arterioles and veins, arterioles and venules that could describe dysfunction. So here's a normal pattern where you can see arterioles and you can see veins and you can see things flowing very nicely. Here's a pattern that he calls stagnant. When you look at the difference between these two, you can see that there's almost nothing in the arterioles. You can see the veins and they look like they're large, and that would imply that things aren't moving very well. And then there are states where you get both a stagnant response in enlarged venules and some arterioles and capillaries that don't fill at all, but some that are actually moving very quickly. So these are the patterns of microcirculatory abnormalities that one can see in patients with sepsis and patients with MODS, and they certainly demonstrate the presence of the microvascular abnormality. The second abnormality to be talked about, though, is this cytopathic hypoxia. This is something that our laboratory has studied quite a bit, and we're certainly not alone in it. And it would imply that the electron transport cascade, shown in this cartoon here, has something going wrong with it. Now, the transport cascade consists of 5 complexes that are linked, that pass electrons delivered from various and sundry different metabolic substrate, ultimately to be delivered to oxygen used to create water, and the energy gradient that develops used to generate high-energy phosphate, ATP. Well, a group Mervyn Singer's group in London has clearly demonstrated abnormalities both in humans and in animal models in complex 1. Complex 2 and 3 tend to be linked to each other, and they're difficult to separate experimentally, but a group in Texas has demonstrated abnormalities in this linkage. Our own work, Rick Levy's work primarily, working in my lab and then on his own, has shown abnormalities in complex 4, cytochrome C oxidase, which I'll talk about more extensively in a moment. And the net effect is a failure to generate ATP, abnormalities of oxygen utilization. As you can probably hear, I'm losing my voice. I hope it'll last through this talk. How did we do the work to demonstrate complex 4 dysfunction? Well, essentially what we did was we made animals septic or subjected them to sham operation, and we isolated mitochondria, and we fed the mitochondria cytochrome C for a specific amount of time in the presence of all kinds of excesses of other substrates, and looked at the kinetics, how much cytochrome C was reduced over time. Now, in looking at enzyme kinetics, the easiest way to look at them, the most beneficial way, is with the so-called Lineweaver-Burke plot, which is essentially an inverse plot of the velocity of the response over the and 1 over the concentration. So you would measure velocity at any given concentration of cytochrome C. And again, it's important to say that this is cytochrome C. That's the variable substrate, not oxygen, but cytochrome C. And do these measurements, do the inverse plot. The advantage of doing it that way is that you can extrapolate this line all the way down to a concentration, to a velocity, 1 over the velocity of 0, which means a velocity that's infinite, and you get to a point where you can talk about the maximum velocity. That is, where this line intersects the x-axis will give you the maximum velocity. So, where it intersects the y-axis will give you the maximum velocity. So again, this is 1 over the maximum velocity, and it will tell you how fast the enzyme can work, a great deal about kinetics. A change in the slope, in this particular case, an increase in the slope, means a decrease in the velocity overall, indicates decreased relaxation velocity at some concentration, but if the Vmax doesn't change, the change in velocity implies inhibition. If there's no change in the Vmax, this is competitive inhibition. It implies that something is binding to the same site where, in this case, the cytochrome C would be binding and competing for it. But ultimately, if you put enough cytochrome C in, you'll reach the maximum velocity and the competition will go away. An alternative would be this, where both the Vmax changes, but the intersection of with the x-axis does not change. So a decrease in maximal velocity with an altered Vmax. This is non-competitive inhibition. It implies something binding to the substrate enzyme complex in a way that inactivates it, something that's locking it into a position. And the final change is where you see both intercepts changing. In that case, again, decreased reduction velocity, but in this case, the change in the x-intercept indicates a difference in the binding site. Something is changing, and this is uncompetitive inhibition. Implies that something is binding to another place on the enzyme, something remote from the actual site of cytochrome C binding, that interferes or limits substrate binding or the reaction kinetics. So again, 3 different types of inhibition, each of which may be at work here. So these are the actual measurements that we did. This is an animal subjected to cecal ligation and puncture model of sepsis. And the red line is the severe sepsis model. This is data accumulated from heart at 48 hours. And what you can see is that both the x-intercept and the y-intercept are changed for the double puncture, implying that this is uncompetitive inhibition. This is a similar plot in the septic liver, and you can again see the same thing, uncompetitive inhibition, change in y-intercept and x-intercept, uncompetitive inhibition. So how do we explain this? Well, the nice thing about cytochrome C oxidase is that it's this lovely heme enzyme, and it has a couple of important centers. There's a copper center where cytochrome C itself will bind, and then there are a couple of heme centers where oxygen can bind, and the electrons are transferred down this protein chain until they get to the oxygen. So what actually happens is that cytochrome C will come in here, it'll donate a couple of electrons, they bind to the copper site, they're then delivered down to oxygen at the heme sites. The oxygen is then used to reduced to water, picking up a couple of protons as it adds electrons. And this works very nicely. Now, when you have uncompetitive inhibition, what you're applying is that something is interfering with the binding of oxygen, but it's interfering you're implying that something is interfering with the kinetics, but not at the place where the cytochrome C is bound. So the cytochrome C is bound up at that copper center. Interfering with oxygen would explain uncompetitive inhibition. So something is going on down here at these heme sites that's interfering with the transfer of electrons that are donated at the copper site. What might those things be? Well, again, uncompetitive inhibition, interfering with something remote site from cytochrome C binding. It might be nitric oxide. It might be carbon monoxide. It might be hydrogen sulfide. All these things have been shown to be increased in sepsis. All these things will interfere with heme binding. All these things could very well be implicated in this particular inhibition. So it's safe to say, based on what I've just shown you, both INSEE's work and my own, that microvascular dysfunction and cytopathic hypoxia do indeed occur, at least in these models of sepsis, and in the case of Jan's work, in human beings with sepsis. But are they pathologic? And that's really the question we have to get to. Well, if this is a pathologic process, there should be some things that absolutely have to happen. The first thing is that ATP should go away. If you're blocking the generation of ATP and something bad is going to happen, it should be because you're depleting ATP. The second thing is that the cells themselves should be damaged and die. If this is pathologic, the cells should be harmed. And organs should fail. Not become dysfunctional, which they do, but fail, which means irreversible dysfunction. Well, what about ATP concentrations? This is again Mervyn Singer's group looking at skeletal muscle ATP in patients with MODS and in controls. And what you can see here, and there's similar data in liver, what you can see here if you look at the survivors at least compared to the controls is that if anything ATP goes up, and it's certainly not depleted. So ATP is not depleted, and this is patients in MODS. If there's no blood supply, there should be necrosis or oncosis. If there's mitochondrial failure, there should also be apoptosis from cytochrome C leaks. There is indeed death in rapid turnover populations via apoptosis, things like the epithelial of the intestinal tract, white cells, lymphocytes. These do indeed show some level of apoptosis that may or may not be accelerated. It's not clear. There is some death of pulmonary epithelial cells, but again, it's pretty hard to figure out exactly what the etiology of that is and whether or not it's really secondary to hypoxemia or failure of oxygen utilization. But there's certainly little apoptosis or necrosis in solid organs. So what we see here is cardiac muscle, and you can see that there's very little difference when you go from baseline to sham to 2CLP. Kidney, again, very little difference. And the final tissue down below, liver, again, some disorganization of the liver structure, but the cells themselves are more or less normal. There is some apoptosis, but it's relatively minor in the overall scheme of things. So there's not massive cell necrosis in severe sepsis or MODS. Well, that's two. Recovery of organ function in survivors is actually the norm. Cardiac endothelial dysfunction, correct, although there may be some residual damage, and we'll get to that in a moment. Fulminant hepatic failure is very rare. The liver has tremendous capacity to repair itself. There is rarely need for long-term dialysis in patients who survive. The exceptions seem to be in the lung, where damage is more permanent, perhaps in neuromuscular activity, where the loss of muscle activity due to perhaps bed rest or immobility is substantial, and cognitive dysfunction in brain tissue, which again doesn't do terribly well when deprived. So it may be reasonable to say that severe sepsis and MODS aren't truly pathological processes. So if they're not truly pathological processes, if this isn't pathology, what's going on? And the question we now need to ask ourselves is not how does organ dysfunction develop, but why does it develop? And that leads us to the key. I would hypothesize the following. Ultimately, what a cell wants to do is live. It doesn't want to die. When severely threatened, that becomes the priority. And in severe stress, all the other priorities may be lost. So the imperative for an organ to function or contribute to the function of the organism as a whole may be lost when its survival, individual cell level, is threatened, and therefore individual cells will limit their activity to that which is essential for survival and viability. That is, maintaining membrane integrity, making sure that sodium is pumped out and potassium is pumped in, etc., etc., etc. And the rest of the things that these cells are programmed to do cease to happen. Well, when we're looking at attempted cell preservation, again, what do we mean? Let's test this hypothesis in a couple of organ systems, heart and liver. So, cardiac dysfunction and sepsis. This is, again, very nice work by Ed Sherwood's group. Ed is leaving Texas and moving to Vanderbilt and Nashville, but he's done some lovely work in this relationship. And the way we best characterize cardiac function or dysfunction is looking at a tangent in a pressure-volume curve to the left ventricular end systolic pressure, end systolic relationship in the pressure-volume curve. So again, you can see where this line is tangent to the end systolic pressure volume. With impaired contractility, what happens is the slope will change from that to that. As the slope goes down, the contractility is impaired. This has been shown to happen, at least in these experimental models of sepsis. This is a little more difficult to do in human beings, although it is possible and has been confirmed. And when you actually quantify this stuff, and we've done this indirectly with excuse me, with cardiac catheters and with some echo, what you can see is that the left ventricular end diastolic pressure volume relationship decreases over time as the sepsis becomes more profound. And this happens in the setting of uncompetitive inhibition of complex 4 and preserved ATP content. So it's impaired contractility, but it looks like it's happening so that ATP doesn't get used up. So an alternative explanation to calling it dysfunction would be say that sepsis results in a state of hypocontractility, uncompetitive complex 4 inhibition, preserved ATP complex, other conditions where this same sort of thing happens and produces a similar result. Well, the answer is yes. After a myocardial infarction, after ischemia and hypoxia and anoxia with revascularization, we all see the phenomenon that we called stunned myocardium, or myocardial hibernation, which is to say that after an MI, the heart doesn't contract properly. for some period of time until it's convinced itself that things are okay and function recovers. Again, we've all seen the state where patients have a fairly large MI. You do an echo, their ejection fraction is 10, 15, 18%. You do the same echo a week later, and they're back up to 35 or 40%. They've recovered substantially. At least part of the loss of function was on the basis of attempting to preserve function. The heart goes to sleep. Cells in sepsis then are deprived of oxygen or cannot use oxygen. Mitochondrial complex 4 does not work properly. Some have hypothesized actually that complex 4 is the sensor that makes cardiac hibernation possible via the O2 binding site, work primarily done by Schumacher's group. And indeed, in the setting of myocardial infarction, hibernation is an adaptive response. During hibernation, myocardium is viable but hypocontractile, and hibernating myocardium downregulates oxygen consumption, energy requirements, and ATP demand to preserve viability. What are the characteristics that define myocardial hibernation in the setting of myocardial infarction? Well, there's a switch in substrate utilization from fatty acids, which heart normally burns, to glucose. There's an upregulation of glucose transporters to make glucose useful. This allows anaerobic respiration, an increase in glycogen deposition. Do these things occur in septic myocardium? Well, the answer is, indeed they do. This is using 18-deoxyglucose uptake in heart, and what you can see is that there's a great increase in glucose uptake and glucose utilization in the heart from the septic animal. Glucose transporters, here looking at the GLUT4 transporter. GLUT1 is the other transporter that works in myocardium. It seems to be unchanged, but the density of the GLUT4 transporter goes up. And the arrow is pointing to these little pink lines, which are glycogen in the PAS staining here, indicate increased glycogen deposition. So all three of the defining characteristics of hibernation do occur in septic myocardium, at least in these animal models. So myocardium in sepsis does appear to hibernate. It's not due to hypoxemia. We've looked. Blood flow is fine. There's plenty of oxygen delivery. It's not due to ischemia. Again, blood flow, if anything, is increased. And again, we've looked. And it's not due to hyperglycemia. Glucose levels are mildly elevated, but not severely elevated. How about something similar in liver? Well, let's define what the liver normally does. It normally neutralizes toxins, those that are developed by the body and those that are put into the body. It produces glucose and other metabolic substrate. It responds to signals from other systems. It's capable of regeneration, which is to say that liver cells that die go away, but the cells that remain can replicate themselves and restore the mass. So, in sepsis, liver cells do not synthesize glucose appropriately, do not clear toxins appropriately, do not respond to extracellular signals appropriately, do not regenerate appropriately, do not synthesize protein appropriately, do not appropriately do the things that affect the organism as a whole, but don't become necrotic or apoptotic. So let's look at each of these in turn. Well, there's a vast literature on gluconeogenesis and sepsis going back to 1974. It's pretty clear that gluconeogenesis, certainly based on substrate availability, is impaired, despite the fact that the animals may be hyperglycemic. Clearing toxins appropriately. Well, again, an endogenous toxin, bilirubin. The defining characteristic of hepatic dysfunction and sepsis, the way we define the disorder, is the presence of hyperbilirubinemia, inability to clear an endogenous toxin. And here you can see it as in one of the descriptions of the MOD syndrome, where bilirubin and perhaps synthetic function, the prothrombin time, are characteristics. Respond to extracellular signals. Well, we've looked at a number of them. I'll give you here the one to IL-6. Normally, IL-6 will provoke activation and DNA binding of a transcription factor called STAT3. What you can see here in the model of septic animals in the liver is that in the area shown by the yellow square, there's severe depletion, blocking of the ability of STAT3 to be activated. in severe sepsis. So indeed, generating extracellular signals is impaired. Regeneration. This again is looking at regeneration using bromouridine. And what you can see here is that in sham operation, the yellow arrows indicate areas where the bromouridine is binding. But when you look at mild CLP, again a lot, but severe sepsis, double puncture CLP, there's virtually no regeneration. And we've done some work that indicates that that this involves a failure to activate cyclin D1. The transcription factor RB needs to be dephosphorylated to be active. It's blocked. It doesn't dephosphorylate. And when all these things are blocked, you don't generate these enzymes, DNA polymerase, thymidine kinase, the things that are essential for DNA replication, and you don't get DNA replication. So again, don't synthesize liver proteins properly. Well, we can look at a whole series of things. These happen to be bile acid transporters, and what you can see is looking at the squares is that the ability to generate these proteins is very severely depleted in the septic animals. So there are a host of other things that are involved. We can safely say that the liver in this setting, in severe sepsis in an animal model, does not appropriately do the things that affect the organism as a whole, but the cells don't become necrotic or massively apoptotic. And it's, I think, logical to conclude that severe sepsis may well be an adaptive state, a response of cells to an insult by attempting to preserve their function. So then the question becomes this. Cells don't die here, but they do become dysfunctional. Patients don't die here either. In general, we can resuscitate people from septic shock if we get there early enough. And that's not where the problem lies. And if this is an adaptive state, the last question we have to ask ourselves is, why don't my patients get better? And the answer lies in the clinical course. So even if the cells do maintain membrane integrity so that they don't die, that still requires energy, which means the mitochondria have to be functional. And for the mitochondria to be functional, there are a bunch of processes that have to happen. And some of the processes that are impaired, the things that we talked about in the liver, are also important in mitochondria. For example, mitochondria-like cells transcribe and regenerate. Mitochondria have their own DNA. They encode 39 different proteins. This DNA has to be replicated if the mitochondria is to remain viable or if it's to reproduce itself. Excuse me. I think what you can see here in looking at, again, at severely septic animals, looking at one particular enzyme encoded by mitochondrial DNA, is that its expression is decreased as we get to severe sepsis. So we've also seen similar findings in liver. It's reflected in protein depletion. The other thing that mitochondria do is they turn over. Mitochondria die on a regular basis, and they're replaced by a process called biogenesis. And failure of biogenesis would deprive a cell of its energy supply and eventually result in cell death. And it appears that biogenesis is impaired in experimental mods. These are stains using mitotracker, a protein that binds, a fluorescent protein that binds to active mitochondria. We mark the mitochondria with a surface antigen called VDAC. The green is VDAC, the red is active mitochondria. Sham operation or mild sepsis, you can see lots of red in there. There are a bunch of active mitochondria. When you get over here to the severe sepsis, there's very little in the way of mitochondrial activity. It is lost. The DNA within the mitochondria are not replicated properly. Something goes on. Both mitochondrial DNA expression and biogenesis are energy requiring. Energy gets depleted eventually. You have to use it to maintain the cell integrity. Both processes will fail. The mitochondria die. They're not replaced. And organ failure then becomes maladaptive. But the initial response may not be. So again, cells do die down here in the chronic critical illness, and so do the patients. So we've hypothesized that severe sepsis provokes a state where molecular oxygen either is not available or can't be used. Cells respond by restricting activity to processes that are essential for survival, membrane integrity, maintenance of the internal milieu. They limit ATP use, and therefore ATP is consumed very slowly. This allows cells to rest, to be in metabolic quiescence, or to hibernate, until either severe sepsis resolves or there's no ATP left. And when there's no ATP left, mitochondrial biogenesis fails in the cell, and the patients ultimately die. On that note, I'll say thank you, and I'll be happy to answer some questions.